Hello everyone, welcome back to the channel. This is the continuation of the embryology class. Today's topic is the embryonic period. Embryonic period extends from the 3rd week to the 8th week. This is the period of organogenesis. It is the time when each of the 3 germ layers, ectoderm, mesoderm, endoderm, gives rise to a number of specific tissues and organs. By the end of the embryonic period, if you see, the main organ systems have been established, rendering the major features of the external body form recognizable by the end of the 2nd month. So in this slide you are saying this is the day 17th. We are having 3 germ layers formed in the 3rd week, that is the ectoderm and endoderm. So this is the endoderm and the intermediate is the mesoderm. So in the middle notochord has formed. So this is the 17th day transverse section of the trilaminar germ disc. So this period extends from, already we have seen what is this period extends from. Anyone? Can you tell? So in this, first we will see the 4th week. What are the changes in the 4th week? So in this 4th week mainly 3 germ layers, they differentiate. Individual differentiation of the 3 germ layers. And formation of folds of the embryo. Still now they are in the form of a disc-like appearance. In this 4th week, foldings of the embryo occur and they, by the way of head folds, tail fold and lateral folds, the embryo assumes the cylindrical shape. In this again one important change is the formation of series of mesodermal somites on each side of the midline. So these 3 changes occur in the 4th week. So in this picture you can see these are the somites on each side of the midline. So these are the somites, but the terms are in this which is used is the older term, primitive segments, which are used older term to identify the somites. This is the dorsum of the embryo, human embryo, 2 mm in length. So now we will see little detail about the somites. Somite period is 20th to 30th day of development. So this again we have the pre-somite period, that is from 15th to the 20th day, and post-somite period after the 30th day. In the post-somite period, mostly the embryo is measured by the CR length. CR length means crown rump length, that is from the cranial end of the embryo to the caudal end of the embryo. Embryo at 32nd day is 5 mm. Up to 55th day it grows by 1 mm each day. Thereafter, yes, you have to tell now thereafter how it grows. You can find out and you can see its interesting feature how it grows after 55th day. So what are you seeing in this picture? So one definition, one term it is given is anencephaly and spina bifida. So these are the defects of the neural tube formation, which we are going to see in this class, which is an important topic. Neural tube, if it is not formed properly, we are going to get these neural tube defects. They are the anencephaly and the spina bifida. So here the cranium is not formed properly, here the spine is not formed properly. Now we will see the neurulation. So first we are going to see the differentiation of the ectodermal layer. The ectodermal layer differentiates into neurectoderm, which will give rise to the neural tube. That process will be called as the neurulation. So what is this neurulation? Neurectodermal cells overlying the notochord, that is thickened to form the medullary plate or neural plate. So under this influence of the notochord, we have seen already in the previous class, primitive streak is the primary organizer and notochord is the secondary organizer. So this notochord induces the development of the neural tube. So under this influence, this ectoderm differentiates into neurectoderm. So this is the formation of the neural tube. First we will see what is that layers. We have already seen this is the ectoderm and this is the endoderm. Now the mesoderm has differentiated into three layers. So this is paraxial mesoderm and intermediate mesoderm and the lateral plate mesoderm. So this is the notochord. Under this influence, so this is the new ectoderm which is forming the thickening first in the beginning. So that is the formation of the starting of the neural. So during the Somite period, the neural plate, which is the thickening of the ectoderm, now forms a gutter known as neural groove. So this neural plate is now forming the neural groove. So this neural groove is concave dorsally and bounded by the raised lateral margins. So these raised lateral margins are called as neural folds. The tips of the neural folds. So these tips of the neural folds contain the cells called neural crust cells. What is the extent of this neural groove? If you see, extends in the midline from the Henson's node, that is the primitive node, to the buccopharyngeal membrane. So in this picture you can appreciate the thickened neural ectoderm, neurectoderm. First it is called neural plate. Now it has formed a groove, that is called neural groove. So these raised margins are called as neural folds. So from this neural folds, tips of the neural folds, neural crust cells develop. The neural folds now fuse dorsally so that the groove is converted into a tube. That process is called as neurulation. If you see, fusion first occur in the cervical region, then extends in both directions. So it is not exactly from craniocaudal. It is starting in the cervical region, then extends cranially and caudally. So that when these folds are fusing and forming the neural tube, there are two openings in the tube, cranially and caudally. They are called as anterior and posterior neuropores. So this anterior neuropore closes at about 20 somite stage or in the middle of the fourth week. Posterior neuropore closes at 25 somite stage, end of the fourth week. So this is a good picture. You are seeing first this is the neural tube formation, neural groove formation. In the neural folds, neural crust cells are formed. So this is the way the neural folds are fusing. As we told, it first fuses in the cervical region, then fuses cranially and caudally. So this neural tube has fused. We have this opening. These openings will be called as anterior and posterior neuropores. So this closes at middle of fourth week and this closes at the end of the fourth week. So this entire picture shows the neurulation. First the formation of neural tube, then the neural groove. The neural folds are fused so that this is the neural tube formation. The edges of the neural folds give rise to the neural crust cells. So these are the neural crust cells which give rise to the derivatives of the neural crust. So the neural tube has formed. What are the derivatives? The cephalic part is enlarged so that they will give rise to the brain by forming three successive brain vesicles, forebrain, midbrain and the hindbrain. Rest of the neural tube persists as the spinal cord. So again this is the formation of the neural tube and the fusion of the neural folds in the order from cephalic, then cranial and caudal, leaving the two openings for in the beginning which closes in the fourth week. So you can appreciate, so if this neural tube is not closing properly, so that is going to give rise to the condition called spina bifida. If the cranial end is not going to close properly, then the brain development and the skull development will be in problem. That will be giving rise to the condition called anencephaly, which we saw in the picture in the beginning of the neural tube formation. I hope now this neural tube formation is clear for you. So the types of the spina bifida are the spina bifida occulta, so that there is no protrusion of the meninges or the spinal cord. That is called spina bifida occulta. When the meninges are protruding, then it is called meningocele. When a part of the spinal cord is also present, then it is called myelomeningocele. So we have seen now the neural tube formation. This entire derivatives of the ectodermal germ layer is given in this one picture. Surface ectoderm, which will give rise to the derivatives of epidermis, hair, nails, sebaceous glands. Apart from that, olfactory epithelium, lens, cornea, and the mouth epithelium. So this mouth epithelium is also giving rise to the anterior pituitary. So that these are the derivatives of surface ectoderm. Neural tube will give rise to the brain, spinal cord, and apart from that it gives rise to the part of the pituitary, that is the posterior pituitary. And also the neural tube gives rise to the retina. Then in the middle at the neural folds, neural crust cells are formed. So the derivatives of neural crust are this. We can see peripheral nervous system, adrenal medulla, melanocytes. So these are some of the derivatives of the neural crust. So students can remember the neural tube gives rise to the central nervous system. Neural crust cells give rise to the peripheral nervous system. The neural crust may be asked as short notes for you. You have to write what are neural crust cells, how they are formed, what are the derivatives. So we have completed the derivatives of the ectoderm. Now we will continue with the differentiation of the intraembryonic mesoderm. So this is what we have already seen in the picture so many times: ectoderm, endoderm, and this is the mesoderm on either side of the notochord, and that is the day 17th trilaminar germ disc. Then further differentiation of the intermediate mesoderm now gives rise to three: paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. So first now we will start with the paraxial mesoderm. What is paraxial mesoderm? How it is derived? It is derived from, we have seen in the previous class, all the three layers are derived from primitive streak. So this paraxial mesoderm is also derived from the primitive node and the rostral part of the primitive streak. It extends cranially from the primitive streak to the precaudal plate and also reaches caudal to the otic vesicle on each side of the hindbrain also. This post-otic part will be forming the bilateral solid cord of cells along the side of the notochord, that is beneath the neural tube. So this part undergoes segmentation, that is post-otic part undergoes segmentation, that is called a series of paired somites. So this is that beginning of the day 19th and day 20th, where the somites are formed from the paraxial mesoderm, only from the post-otic part of the paraxial mesoderm. The rostral to the otic vesicle, that is preotic, they remain unsegmented. The students should note that this part is unsegmented, they underlie the developing brain. Only post-otic parts undergoes segmentation and forms the somites. So this preotic part develops the base of the skull and also the muscles of the striated muscles of the head region. So this another name is this metameres between the 20th to the 30th day. The segmentation is extending in the craniocaudal direction. First pair of somites appear just caudal to the tip of the notochord. By the end of the first month, if you see 44 pairs of somites are formed. Somites may be asked as short notes. You have to write what are somites, how they are formed, when they are formed, and the numbers. So we call this 4th week period of development as the somite period. So in this picture you can see approximate age, that is by days. So this first 20th day 1 to 4, then 21 4 to 7. Like this, we are giving the number of somites correlated to the approximate age. By this number of somites, we can calculate the approximate age of the embryo also. So what are the 44? 4 occipital, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 8 to 10 coccygeal. So what are the derivatives? What these somites will give rise to? Cranial most 4 to 5 pairs, they are called occipital somites. They are going to give rise to the formation of the base of the skull. If you see again, each somite is developing a cavity called myocele, so that this is somite is divided into two parts, ventromedial part that is called sclerotome and dorsolateral part that is called dermomyotome. What is this sclerotome? So the sclerotome is the part which is going to give rise to the skeletal part. So it migrates around the notochord and the neural tube to give rise to the primitive vertebrae. So we have seen in this slide, we have seen these two parts of the somites, sclerotome and dermomyotome. First, the sclerotome, what is it giving rise to? Primitive vertebrae. So you can see in this picture, so this is the, if you consider this as one somite, so this part is called sclerotome and the other part is called dermomyotome. So this sclerotome migrate around this notochord and the neural tube, so that it will form around the neural tube the primitive vertebrae. This dermomyotome will give rise to muscle and the dermis of the skin. So this dermomyotome divided into two parts, lateral dermal plate and medial muscle plate. Dermal plate forms the dermis of the skin and subcutaneous tissue. Myotome gives rise to the skeletal muscle of the body. All striated muscles of the trunk, limbs, muscles of the tongue, diaphragm, they are derived from this myotome part of the somites. Sclerotome portion of the somite gives rise to the development of vertebral column and the ribs. So we have completed the somite formation in detail, including the derivatives. Now we are going to see the intermediate mesoderm. It will give rise to the nephric system and the sex glands. So in the cervical and upper thoracic region, it is segmented, and in the caudal part, it is not segmented. It is called nephrogenic cord. So it is going to give rise to the urinary system and part of the reproductive system. What is this lateral plate mesoderm? It is derived from the middle of this primitive streak, which is unsegmented. So this lateral plate, you have to imagine it is reaching around the peripheral margin of the embryo and also cephalic to the buccopharyngeal membrane. So this, when the somites are forming, simultaneously in the lateral plate, series of clefts are appearing, so that these clefts fuse with each other. Inverted U-shaped tubular passage, this will be called as intraembryonic coelom, forms in the lateral plate mesoderm. So this is what we are describing. You can see in the lateral plate cavities appear, so that when they fuse it will be called as intraembryonic coelom. Students should appreciate this is extraembryonic coelom. Now this is the formation of intraembryonic coelom. So simultaneously when the neural tube is forming, the somites are forming, lateral plate is forming the intraembryonic coelom, which is now continuous with the extraembryonic coelom. So you can see this part of this along with the ectoderm, this part of the lateral plate mesoderm will be called as somatopleuric intraembryonic mesoderm. the part adjoining to the endoderm, that will be called splanchnopleuric intraembryonic mesoderm. So the students should understand, in the previous third week we have seen somatopleuric extraembryonic and intraembryonic. Here now in this we are seeing the intraembryonic. So this picture shows the formation of the somites. So this is the paraxial mesoderm which has given rise to the somites. So this is the intermediate mesoderm. This is the lateral plate mesoderm which has formed the intraembryonic coelom, which in the future will give rise to the pericardial cavity, all the pleural cavity, peritoneal cavity that will be formed from the intraembryonic coelom, and this is the communication with the extraembryonic coelom. You can appreciate there is a one part still cephalic to this, there is, which is not divided. So what is that part? So the intraembryonic coelom, already I have told, divides the lateral plate into two layers. Somatic layer with the overlying ectoderm will be called as somatopleure. So that is intraembryonic. Splanchnic layer with the endoderm will be called as splanchnopleure. So again we are seeing clearly in this picture intraembryonic coelom. What is this will be called? Layer will be called somatopleure, and this together will be called as splanchnopleure. So again the same, along with the amniotic cavity and yolk sac is given in these pictures. So we are seeing now three layers. Three layers are dividing into three parts: paraxial, intermediate and lateral plate. In this we are seeing this clefts appear, clefts have fused, so that intraembryonic coelom appear, which is now continuous with the intraembryonic body cavity. Now it is formed. So as I was telling, this is the undivided part. That part is called as septum transversum. It may be the short notes for you. What is this? This is the horseshoe shaped mesodermal plate adjoining the pericardial sac and the coelomic ducts. That is the definition, called as septum transversum. So we will see in the coming embryology classes, we will see what are the derivatives of septum transversum also. But in this class, first you should understand what is septum transversum. So what are the layers of somatopleuric layer? Somatopleuric layer give rise to parietal layer of all the serous sacs, dermis and subcutaneous tissues of the body wall, and skeletal elements of the limb buds. Otherwise, you are now dissecting this upper limb and lower limb. So all the bones in the limbs, that is derived from this somatopleuric layer of lateral plate. But the muscles of the limbs are derived from, yes, myotome. Splanchnopluric layer of intraembryonic, that will give rise to the visceral. We will be seeing in detail this pericardium, pleura and the peritoneum. We will be seeing parietal layer and visceral layer. Parietal layer is given by somatopleuric and visceral layer is given by splanchnopluric. So this splanchnopluric layer gives rise to the all the muscle and connective tissues of the gut, respiratory tubes and the heart. So we have seen in elaborately important topics in this class. One we have seen is the neural tube formation. Again we have seen the differentiation of the intraembryonic mesoderm. We have seen the formation of the somites in detail in this class, and in the lateral plate we have seen the coelomic cavity has formed. We have seen the derivatives of somites, derivatives of intraembryonic mesoderm, derivatives of somatopleure, splanchnoplure also in this class. So which completes the important stages in this third week, that is starting in the embryonic period. We have seen neural tube formation, somite formation is the fourth week, and now we have seen the formation of intraembryonic coelom also. In the fourth week, foldings of embryo also occur, which we will see in the next video. All of you, those who have not subscribed to the channel, do subscribe now, so that we will see the fourth week development in the next video. I hope you should have understood these important topics, new relation and somites. Thank you.